Okay, well, great. Welcome everyone to what will perhaps be the first of many conferences on neuroscience in the everyday world. I wish I could say I'm actually recording my brain activity live while welcoming you all, but just not yet. I'm just showing this off. Next year, however, we'll be watching images of my brain activity during the start of this conference. So we are all here today because of our interest in understanding how the brain functions in the everyday world environment, and how it enables us to interact and perceive with the world around us, and how disorders of the brain affect our ability to actually engage in the world. So we all represent many different disciplines, all focused on this problem from very diverse perspectives. We're engineers developing hardware, we're computer scientists who are developing analysis methods, we're cognitive scientists, who investigate the healthy brain, and clinical neuroscientists, interested in those disorders and how that affects our ability to engage the world. So our goal over the next day and a half is to broaden understanding of the diverse aspects of this new field, neuroscience in the everyday world. So we have many lectures representing these diverse aspects of the field, but really the benefit for all of us is going to come from the discussions we have, right? So we want to have ample time for discussion after every lecture, and we have the coffee breaks and the lunch and the poster sessions. So we want to have just lots of good engagement. We all really are coming at this from many different perspectives, and I think there's a great deal we can learn from each other. So I'm a physicist by background and a professor of biomedical engineering. I've been developing FNIRS technology for over 2 decades. And so while my group in 1998 was one of the first to build an imaging FNIRS systems, we were quite late to developing wearable systems. It was only in 2016 that we first started to build a wearable FNIRS system. In 2020, we were very fortunate. We got a nice NIH Brain Initiative grant to build this system. So this is coming online now. Maybe later today or tomorrow, I'll actually turn it on so you can see the signals. It's 200 optodes, 56 sources, 144 detectors. Absolutely amazing, but it's a huge amount of data. And I can collect this data while presenting to you. But I'm really kind of just beginning to think about how the heck are we going to analyze this data? How are we going to design experiments so we can actually try to tease apart the functioning brain in natural environments, right? I really don't know. Right, so that was really the impetus to try to organize this meeting to bring together, all of you. So for me personally, to help me understand what am I going to do with this, but I think each of us has our own motivation for being here today. So we're also very fortunate. The last year, Swati and I got to know the Tian Chao and Chrissy Chen Institute, who their foundation shares very common interests with us. And they encourage us to organize this conference, to push this field forward, to help accelerate it. So they helped us co-organize this, very gracious in the support, bringing all of us together. So Swati and I are very grateful to them. We're also very grateful to Boston University and the Office of Research, led by the Vice President of Research, Gloria Waters, who's joining us at this conference, and their support of the centers at the university. It's through Swati's Center for Brain Recovery and my Center for Neurophotonics that we bring together diverse groups of faculty who work on similar problems. And through these centers, we're also able to help support this conference and these interactions. With that, I'll hand it over to Swati Kiran, who has a few more remarks.
Thanks for that great introduction, David. And wearing that, David is in the middle of data collection this week, so he's all about the cap, and we're so excited. I just have a few logistical things to go over really, really quick. You can visit, there are lots of seats in the front. Please come in, and we'll manage the overflow room as well. As David said, we're really fortunate to have the support of the Chen Institute that's co-hosting this conference with us. This is yesterday's news. We had over 328 registrations. I'm still fielding registrations this morning. I'm hoping to get them the Zoom link in as we speak. And one of the things that the foundation, the Institute program officers encouraged us to do was to have the conference to be able to be streamable. And we're really happy we're able to do that with the support of Boston University, because see how far we've reached. This conference is generating a lot of excitement. I'm really excited to see some of the virtual registrants, and this is really not all the map, because this map was made on Sunday and we have had so many more registrations till then. The other thing I just want to point out is they've also, the Chen Institute also was very generous in their funding for student travel awardees. And so I just want to take a minute to recognize all of the student travel awardees. If they could just stand up as I read their name. They've actually come from various parts of the world, Ireland, and I'm trying to remember all the other places. So Michelle Goodrick is here, Yipit Topoglu is here, Katerina Wisieska, Kasia. I think she's supposed to be here. She's on her way from Ireland or something. Kevin Ramirez Chavez is here. Adrian Curtin is here. Suman Byun couldn't make it, but I hope his professor can make it. Leila Kouara is here. Andrew Hagan is here. Condell Eastman is here. And Ike Middle is here. So thank you for coming. They are going to get together over the course of today, and tomorrow at the end of the conference, they will do a panel summary of what they think are the most exciting issues in this field, and what are the next set of issues that people, researchers, young researchers like them should be addressing. As you all guessed, this is a very nascent topic. We're actually astounded at the response to it, because it's such a new field, and it's taking a lot of interdisciplinary folks together. So it's time to share our work, but it's also time to share ideas and challenges. So we're looking forward to that. Please stay till tomorrow and listen to their summary. Thank you very much for coming. And please feel free to reach out. But maybe one of the things that we can just get people going here is just, why did you come to this meeting? And what's interesting you? Just a few ideas here, just so that we can also think about what topics to focus on in the panel discussion. So things that generated your interest to register and come to BU on the train and the car, which is not an easy thing to do at 8 o'clock in the morning on a move-in week when everybody's moving in. So thank you for coming, but maybe a few people can just chime in and say why they felt like joining this meeting. Yeah.
Hello, I'm Alex. I'm a first year's master's student at BU. So I came to this conference because I have a background in psychology and neuroscience, and I found it interesting with some of the topics of conversation. I have an interest in neuroregenerative medicine, and so this talk and talking about the interface with the brain and AI was also very interesting to me. That's why I'm here today.
Great, welcome. Anybody else? Yeah.
My name is Michael Bell. I'm a retired professor of biology at BU. I'm a neuroscientist. I have 3 other neuroscientists, retirees, who have had careers in this country over the last 50 years. And we've all been talking online about consciousness recently. I think at a point in life that I'm at, you start to think back about what is the brain and what does it do? And this topic of the everyday aspect of neuroscience is very relevant to that for me at this stage in my life. That's why I came to this.
So are you hoping that at some point we'll be able to study the brain as we're thinking and in conscious?
That maybe you'll tell us what consciousness is.
Yes.
Hi, this is Pepe Dan, coming from Japan. I think, as far as I remember, I was the first one to put the word every day, which is associated with neuroscience. It was 2003, which was about an apple peeling paper assessed by functional neuro. However, at that time, nobody was interested in everyday neuroscience. So I started too early. But well, nowadays, I was really amazed to find that so many people are coming to this conference, and probably this is the right time that we can actually do everyday neuroscience. together. So I'd like to witness this moment. That's why I came here.
Thank you, Dan. Pepe, that was amazing. Thank you for saying that. And I think you're going to feel that energy from many of the speakers today, because I think we're all thinking it's time now. We have the tools. We know how to be looking at this. Let's give it a shot. That was really well said. Any other comments? Students? Any other student? Yeah.
Other students at Colorado State University, and we have done a variety of things, but I'm kind of more in the neuromechanics. So I come from a biomechanics movement background, and we're integrating some FNIRs. We've traditionally done some MRI stuff as well. But I think just hearing about all the different things that people are doing with this type of neuroimaging, and different things like, hey, how are you processing this versus this? And I think we're getting to the point where we're getting some more standards in the industry of how we're doing these things, and I think that's going to be really cool to hear about.
Yeah. Yeah, I hope we get to that point. I'm not sure we're ready yet with standards in terms of these topics, but that is exactly the kind of discussion we're hoping to have tomorrow, is what should we be thinking about. All right, well, thank you for coming, for all the reasons that you decided to choose your time today to spend here. We appreciate it, and we're hoping for a great discussion. I think we're ready, Simona, to get started, and I'll hand it over to David and Joy.
Thank you. Thank you, Swati. So just remind everyone the schedule. So we have first a keynote talk from Joy Hirsch, and then we'll have our first session that's focused on technology. That will be followed by lunch and the poster session. So we have 2.5 hours there for lunch and poster session. So really use that time to get to know everyone here and have that discussion. Okay, so it's my great pleasure to introduce our first keynote today is Joy Hirsch. Actually, it's quite funny. We were actually, many of us, well, Joy and Alex and several of us were actually here 10 months ago because we had the Society for FNIRS conference here in Boston just 10 months ago. So it feels a little bit like deja vu. But she was absolutely the right person to have come and speak to us because of her background of functional brain imaging and moving that into the everyday world. So she is the Elizabeth Mayers and House Jamieson Professor of Psychiatry and Comparative Medicine and Neuroscience at Yale School of Medicine, and she's the director of the Brain Function Laboratory there. She's also a professor of neuroscience in the Department of Medical Physics and Biomedical Engineering at the University College of London. So the overarching goal of her research is to understand the neural mechanisms that underlie live, interactive social behaviors between individuals. Earlier in her career, she founded and directed the fMRI Research Center at Columbia University, dedicated to investigations of the brain and translations aimed at understanding the biological mechanisms of neurological and psychiatric disorders. But more recently, and really relevant to this conference, in an effort to expand neuroimaging to live dyadic interactions, as opposed to single brain processes as investigated by fMRI, she really envisioned a new two-person imaging system based on head-mounted detectors using functional near-infrared spectroscopy, much like what you see me wearing here, right? And so she's been really engaged in developing a number of new research paradigms to investigate these neural systems of the engaging interactions between two humans, right? So very relevant to today's conference. So please join me in welcoming Joy Hirsch to the stage.
Thank you, David. Thank you for a very generous and kind introduction, and for all of you for being here. I feel so humbled and so honored to be the person who is here today to kick off what I think is a major landmark event in the history of our science, of our neuroscience, particularly the neuroscience looking at this whole new domain of live interactive behaviors that we sort of lump together in this description of the everyday world. And thank you, Ipadan, for coming up with this idea of science in the everyday world. So I've titled my talk, Wild Type Neuroscience, just because I like it, because it's kind of adventurous. It's sort of like out there. And I think that that's the way we have to start thinking about this new science. And I'm going to try to lay out a framework today of challenges, progress, and future directions. And I was putting together this talk. I was humbled by the fact that we know so little, that we're just at the beginning. And I want to share that with you in the spirit of this is a rough draft. This is a template. This is the beginnings, and it's up to us to develop it. Before I go on, however, I want to shout out to the organizers of this conference, David Boaz, Swathi Kiran, who was one of the organizers, and also Alex von Lumen. Also the Tien Shou and Chrissy Chen Institute, the funders who believed in us, who said, go for it, let's go for something different. And these people deserve a tremendous amount of credit. And here we are. We're at the start of a new frontier, looking up at this enormous rock. What a climb we have in front of us, but I like this analogy very much. And I would like to think that in a few years from now, maybe not too many years from now, that we'll begin to look at this function again, but with a little bit different metric. We'll have project the growth of the new science, maybe number of publications, maybe amount of funding, maybe amount of impact we have on science over time. And so I would like to start this with the assumption that we have a climb, a real trajectory, and some real opportunities ahead of us. So let's just start with a very simple idea that we have a domain of well understood neuroscience, of which we have collected over the last, say, 2-3 decades. And these are behaviors that we understand the neural circuitry associated with, that are come from well-defined laboratory behaviors. We've used fMRI, a little bit of FNIRS, psychophysics, EEG, MEG. We know a lot about the brain when our brains are in solo mode. But there's this whole huge domain, which is why we are here today, to address a poorly understood neuroscience of behaviors in the everyday world. Like what David said, what is my brain doing when I'm standing here wearing this thing, talking to you? What is my brain doing when I'm thinking? What is my brain doing when I'm making decisions out there in the real world? This domain of poorly understood behaviors outside of the laboratory is a vast amount of material, and it might even include consciousness, although I've learned to use that word very cautiously. Nonetheless, one might include topics such as that. I like to think of this as our brains in interaction, and that interaction is not only the interaction between each other, but interactions with the real world, interactions with circumstances, and so on. So it's our brain in action. So let's just start with identifying grand challenges. And I've identified 4 grand challenges, which really sort of orient the topic which I'm going to present to you in this talk. First, what are our new set of questions? What do we need to know that we don't already know? Second, how are we going to study them? And this is huge. This is huge. What technology do we need in order to study these questions of behavior that's out there and about in the real world? 3, why do we want to know the answers to these questions? Why are they important? How does this improve how we think about our brains, ourselves, and even medical applications? Why are these important? And 4, lastly, how in the world are we going to fund these organizations? We're not going anywhere unless we can convince the NIH and other funding organizations that what we are doing is important, it's necessary to fund it, and that we are going to be fundamental to future directions in neuroscience. With all of this in mind, there's one grand challenge that we all have to keep in mind, that our overarching goal is to understand neurocorrelates of behavior, in this case, a wide range of behaviors that exist outside the laboratory in the everyday world. So here we go. Now, these 4 grand challenges are intermixed in my talk. I actually tried to do, sort of an organized talk where we do one grand challenge and then the next one and then the next one and then the next one. It didn't work so well. Just let me mush it up a little bit. It's kind of mushed together. 1 and 2 and 3, they kind of go together and I'll kind of pull them out. But just keep in mind, you keep it organized. There's 4 grand challenges and we'll kind of address them in combined ways. Okay, so here we go. First of all, does it matter? Does it matter that the brain is in an interactive mode versus a solo mode? That is, how good is the proxy for behavior that we've studied in solo and non-interactive mode? Well, we can start with a hypothesis that our philosophers have given us, and this is the interactive brain hypothesis. And they, without the benefit of any data, have suggested that yes, that the brain in interaction is different than the brain in solo mode. And others in our field, Leonard Schielbach and Thalia Wheatley and others that have thought about this topic have indeed suggested that interaction is an important goal and the brain actually indeed should be different in interactive and solo mode. Well, we did a little test early on to directly test this hypothesis. And I share this with you here as a sort of a foundational touch point where we have 2 people sitting across from each other in conversation that is interactive. And we have the control experiment where they have the same kind of conversation, people talking and listening, but without any interaction. Now, the bottom line is that it matters a whole lot whether there's interaction in a conversation. Particularly in the posterior aspects of the brain, those areas of the brain that in this case have been referred to as Wernicke's area because it's a language task, that in the non-interactive condition, the Wernicke's area is just simply kind of just doing what it does, just gathering information and interpreting it. But in the interactive condition, it is much more active. This was an early experiment that we did that encouraged us to continue thinking about the brain in interaction and how that would compare without an interactive condition. So this is kind of a starting point. Yes, it matters that the brain is out and about in interaction. So now the next few slides, I'm going to suggest that we might use this idea to identify some future areas. I'm talking about Grand Challenge 1. What are the new questions that we could ask if we really wanted to study interaction in ways that hasn't been studied before? So every dyad in the world, a dyad meaning 2 people, every dyad in the world since the beginning of time probably has experienced conflict. And if not dyads, families, communities, institutions, governments, conflict. Conflict is a part of interactions of who we are as humans. So here's a little foundational study that I think might lead to some ideas about some future directions, where 2 people were in a conversation, or conversations. The conversations were either topics where they agreed or they disagreed, okay? So we compare these individuals, the activity associated with agreement and disagreement. And in red, we see activity associated with disagreement. And in blue, we see the activity associated with agreement. And we see that the brain is very, very different under conditions of disagreement, all that red stuff, frontal lobe stuff, you can imagine what that's all about. And the much more peaceful, more sensory related blue are the agreement. Okay, there's a lot that can be said about that study, but I'm going to go on because I think what this does is suggest that there's a whole new domain out there. You know, that big circle, domain of questions that we haven't addressed yet. It has to do with interpersonal conflict. There's ideas of competition, there's ideas of collaboration, social behaviors like avoiding, accommodating, compromise. All of these things are dyadic behaviors that are not understood, that perhaps we, with our new methodologies, with ideas of studying human behavior that is untethered to laboratory conditions in the way that we've traditionally done it, may have a whole new arena ahead of us. Okay, that's one idea for a topic in that big circle. Another idea is that we know that if one is having a pro-social conversation with someone who is very different than we are, that perhaps it requires a little bit more effort. But we don't know really what's going on in our brains when we talk to people who are outside of our socioeconomic domain. So we ran a quick study to look at that question. The answer is yes, indeed, just as your grandmother might have told you, that it matters a lot whether you're talking to somebody who is part of your homogeneous circle or someone who is outside of it. Well, I think that this idea also provides a kind of inspiration for future directions for us who are interested in science outside the laboratory and in the real world. Could studies such as these provide a way of informing progress and strategies for our DEI objectives, the diversity, inclusion, and equity objectives that are so important to the way that we conduct our institutions of higher education, businesses, and in a world where we are making huge efforts to be inclusive. Could our science contribute to a neurobiology that could inform how we could improve progress and behavior changes with respect to DEI objectives? Just put it out there as a possible and important area for future directions. Another area, as our society ages and more of our care of each other occurs outside of a hospital environment, it occurs in the home, in a home care setting. More and more people are involved in caregiving, and the interactions that go on in caregiving are not well understood at all. In a little study that we did, it was sort of a proof of principle study, says yes, that when one is one is involved in caregiving interactions, that these caregiving interactions actually do employ very specific parts of the executive brain, that this is a very tractable and measurable type of dyadic interaction that is accessible with two-person type studies. So the question is the neurobiology of caretaking a potential new topic for investigations outside the domain of the classical kind of studies that we've done in the laboratory? Now, here's one that I have no example for, but recently, earlier this year, in May 2023, our Surgeon General posted an advisory that has raised a great deal of alarm about loneliness in our society. And he suggested that the disconnectedness that people felt was related to at least increasing the rate of stroke or heart rate and dementia by about 30%. And that this was something that we as a society, and particularly neuroscientists, should be engaged in thinking about how to study. So I would lay out a couple of possible future directions that might address this issue. I think that it might be very possible to understand the therapeutic role of connections between canine companions, for example. This is a study I would love to do. I have I have applied to the NIH a couple times for this study. It has not been successful. But I would love to do that study. Another one is the whole domain of assistive robots, or robots that could be used and applied as companions. Do we have abilities in our community with our two-person dyadic interactions to address some of these new questions? So, okay, so these questions are kind of cool, and there's a lot of them. And for all the ones I can think of here, I'm sure all of you out there can think of more. But what we have to avoid is a kind of what I like to refer to as just gee whiz science. Let's just do stuff and see what happens. In order for this to be successful, we have to do a deeper dive as we look for the fundamental mechanisms of dyadic behaviors in the everyday. We need to develop a theoretical framework. And so to do that, we're going to have to dig a little bit more deeply. And one of the first parts of that is how we acquire our data and what our paradigms are. And this is huge part of our uphill climb as we look up at that big rock and say, how are we going to get there to build this new science of the everyday world? What I want to tell you about in this slide is the system that we use. And first, I just want to point out what its major limitation is that I'm still in the laboratory, guys. Here I am advertising that we should get our science out there in the real world, that we should be doing these wearables. And what I'm talking about is science that's still done in the laboratory, but it's done very differently than conventional laboratory science is. We have two people sitting across from each other. I call this suite a social interaction suite, or we lovingly refer to it as Big Sis. This is the big system as opposed to the little system, and SIS for social international suite. So this is SIS. And SIS has some very interesting features. First, smart glass. And if you can see, I need a pointer. Put on a pointer. Wait, I'm not telling, right? Right there. See that glass? You can barely see. Yeah. This thing right there, that's a smart glass, pretty cool thing. Because what that is we run a current through that glass, and the glass is like a window. You turn it on, the current goes through, and you turn it off. I first found that glass, or found a version of it, in the dressing room of the Prada store in the South Village in New York. You know, you turn it on, the dressing room is open, you turn it off, the dressing room. It's a really cool thing. But anyway, so this is stuff you can buy. You can buy it on Amazon, bring it into the lab. And what this glass does is it allows us to turn the viewing on and turn it off, turn it on and turn it off. It's a very important part of these experimental paradigms. We have dual head F-nears, of course. We have dual eye tracking. The eye tracking is surface mounted, so we know exactly where the eyes are at all the time. We have dual head EEG. We're doing EEG simultaneously with F-nears most of the time. We have dual facial classification cameras, so we're doing facial action units almost all the time on our individuals. We have dual response dials, so people can dial in the amplitude of their emotional or affective responses as necessary. We have paradigm control computers, that everything is synchronized. So all the data streams are synchronized, and computers are, of course, managing that. We have auditory recordings and microphones. All this stuff is hanging from the ceiling. Actually, this was a sidebar here. This was a renovation from COVID. We had to have more space, and I couldn't have stuff on the floor because we needed more space, because that's what the officers about safety told us we had to do. So we just hung it off of the ceiling. It's pretty cool. And I love it. Guys, I love the design because that's occupational hazard. I love the design because we have all this stuff hanging, and it allows us to put a lot more in this social interaction suite than what we would normally. Of course, we have data management and analyses pipelines for all this stuff. physiological measurements. We're measuring skin conductance, respiration, and so on all the time. And multiple experimental paradigms that explore these dyadic behaviors. And this is something that we'll talk a lot more about in this talk and through this conference, I think. But David was absolutely right. This are experimental paradigms and the data analysis techniques that are really essential to develop to take us over the top. Anyway, so this is full disclosure. This is how we are discovering these questions. One of the really important things I think that to take away about this instrumentation is how multimodal it is. And one of the things that we can do with our FNIRS technology, because we do not have the inconvenience of the high magnetic field, is that we can integrate these multiple modes of data streams with the neural data that we have, and it is a major advantage for all of us. And I'll show you some examples of that as we continue on. As part of my deep dive, I want to focus on a particular kind of stimulus, because I think, and that's the face, of course, the face. I think the face, because it is the most compelling of the social stimuli that we have, and what we need to learn about dyadic interactions, we can learn from how the brain processes faces. I actually love this slide. I used to have a slide where I took a lot of faces. You can get them off the internet, get Getty images and so on. And always somebody comes to me and says, aren't you worried about being sued because you're using an image of some person who hasn't given you permission to use their image? And so what I did is I went to my iPhone and I'm taking advantage of all of my friends and family. This is me, these are my people, and none of them have given permission. Tough bananas. And, so anyway, I think that it's important to ask this question now with faces. Does it matter that the face in your real world is real, or is it simulated? So is this the same as this? And we can do a very simple experiment, again, using CIS, comparing it over here, just simple gaze at real faces versus simple gaze, in this case, a dynamic video. And this is the paradigm that we use. And this is really, really important, because I actually don't know exactly how we're going to develop paradigms for studying these types of interactions in the real world, in everyday life. I don't know quite how to do it, but I'm hoping that by the time we get through this next day and a half, maybe some of us will have some better ideas than what we have. This is what we do now. This is a time series. It's 180 seconds, so 3 minutes. And this 3 minutes, we have 30-second epics. We have an activity block and we have a rest block. So this is a block design: activity, rest, activity, rest, activity, rest. The block design is divided so that the task part of it has 3 segments. And those 3 segments are 3 seconds each, and those are the face viewing segments. So we look at a real face for 3 seconds, and then the smart glass turns off, and it's a rest. So it's 3 seconds on, 3 seconds off, 3 seconds on, 3 seconds off, 3 seconds on, 3 seconds off, and then a full rest for the signal. So this is a standard block design for analysis. The most standard way is to treat this whole thing as a block of 15 seconds of task, and we just mush them all together. So we've got an on task of 15 seconds, an off task of 15 seconds, and so on. Now, there's several advantages to this. One is the human factors advantage. One is that you can't stare at somebody for 15 seconds. So if you're looking at someone's face and there's an interaction going on, you don't want to look at that person for about 15 seconds. You look away, right? So, and we're dealing with Yale undergraduates, and they just start laughing at each other, and that doesn't help in experimental design either. So the other thing is that we combined EEG. And EEG needs lots of different triggered events. So this is a lot easier to use, a lot more advantageous to have an event, a disrupted event system like this when we're combining both eye tracking or facial classifications or EEG. So I'll show you a little bit more as we go through this. But this experimental paradigm is really important. Now, the answer to the question, yes, it matters a whole lot, whether the face is real or whether it's a dynamic video. And in particular, it matters with respect to areas. This is the right side of the brain, the supramarginal gyrus, superior temporal gyrus, a simple contrast. And this is a result that's reasonably old result now, but it's been replicated many, many times. We've also had a bit of fun comparing real gaze with robot gaze. This is Maki. Maki's quite human-like, actually. Eyes blink, head moves, head nods, and so on. But you can't fool the human brain. And the human brain knows a whole lot about processing that has to do with the real human, as opposed to, in this case, the robot. And so here we begin to develop a theoretical framework. What really is so different about the real face that's not characteristic of the simulated face? And it is indeed that we have additional activity to process real faces in what I like to call, in this case, the lateral visual system, consisting of superior temporal gyrus and supramarginal gyrus. So this area is also known as a nexus for social activity. Carter and Hoytel, 2013, identified this area using functional imaging studies of having people just observe social behaviors. And this area was also active. So that's not too surprising. So let's dive a little bit deeper into this, again, looking for fundamental mechanisms. What is so special about this real life face? What makes this real life face so special? Well, I'm going to outline 3 hypotheses. And the reason for these 3 hypotheses is first to learn to partly answer the question, what's so special about the real face? But partly I want to use these 3 hypotheses as examples of how we use FNIRS in combination with other variables that illuminate our understanding of the brain. So the first hypothesis is that the real face effect is due to eye-to-eye contact. Well, eye-to-eye contact is something that's pretty easy to study in the CIS paradigm that I outlined. We know where the eyes are of each subject, and we can identify an eye box, and we can determine when both eyes are in each other's eye box, and when they are for 10 frames, which is 0.83 seconds with our eye tracker, the computer codes that as an eye-to-eye contact event. So now we go back and we do exactly the same experiment again, eye gaze versus eye gaze with real faces, with this case, the dynamic video. And we do our standard contrast analysis and we replicate our previous results, but we also add this dorsal system associated with what we hypothesized was due to the eye contact part of the task. To analyze that in more detail, we took the eye-to-eye contact as a covariate, and we used the covariate on the main effect and identified or amplified. That analysis amplifies the effect of the eye contact, relating it to both the dorsal stream and the dorsal lateral frontal cortex. So eye contact is a major part of the real face effect. And in fact, very consistent with hypothesis 1, we can begin to elaborate our model. This is the old ventral stream model, well known from the old studies from fMRI face processing. This is the new lateral stream, the real face effect. And now we've shown additional effects due to eye-to-eye contact. Hypothesis 2, that the real face effect is due to continuous activation of the face system. This gives me an opportunity to show you some examples of using simultaneous EEG and FNIRS to probe some of this activity. So hypothesis 2. So here, I'm going to show you some EEG data with signals during the real phase and also the robot phase. First, the robot. So what I'm showing you here is the alpha band. The alpha band is just a little bit higher frequency than the theta band, 8 to 12 hertz. And we see activity in the lateral. We're using brainstorm here, so we've localized the signals to the lateral stream and the dorsal stream, the two areas that I just showed you. And you see the first 250 milliseconds, the transient occurs and it falls, and then not much is happening. Not much is happening in the robot. But what do you think happens with the real human? Da-da-da-dun-da-dun, here we go. The same stuff, lateral stream, dorsal stream, same areas. We see the transient at 250 milliseconds, and then look at the continued processing, continued out 1500 milliseconds and beyond. And this is an effect that we see over and over again. It is as if the real face, probably we think the micro movements of the face are stimulating continued processing over time. So this is consistent then with hypothesis 2, that yes, there seems to be continuous processing. Now, the question is this due to the micro movements of the face? Well, this is hypothesis 3, that maybe indeed this is due to the little movements, the eye twitch, the tiny little movements of the face that you wouldn't pay that much attention to, but are communicative. And this study was designed to test that hypothesis. And I also include this in this suggestion of hypotheses about what's different about the real face, because it's also an opportunity to show you how one can use facial classifications as a stimulus. If you're out there in the real world and you're looking at faces, you better know a lot about that face that you're seeing. So one way to do this is to have cameras that classify the face. We can look at them, look at the facial action units, process the facial action units, and have a definition of this face, and then look at how the brain processes it. So here we go. This is a very special kind of dyad. This dyad, these two people are doing two different things. One is the movie watcher, one's the face watcher. So she's just the face watcher. Her job is just to watch the face of this guy. This guy here is not looking at her at all. He's only looking at movies, and these movies are funny little movies. They're like 3-second clips. Some of them are funny, some of them are creepy, some of them, like worms and stuff like that is awful. And some of them, anything that's just is designed to get a facial expression. I'm avoiding the word emotion because real scientists have that word all locked up. These are just ways of generating affect. So we've got the movie watcher and the face watcher, and that's just what I said. Okay, so then the timing kind of goes like this. This is an example again of two people sitting across from each other with a smart glass. The smart glass is either open or the smart glass is closed. When the smart glass is open, the watching is going on. She watches his face, he watches the movie and generates facial expressions. When the smart glass is closed, they're both resting, looking straight ahead, but they both use their dials that they have to rate their level of emotional experience, his with the movie and hers with his face. Okay, so here we go. So the first thing is do the affect, the emotional expressions communicate from one person to another. So this is his ratings and that's her ratings of affect. And you see there's a very good correlation. This just suggests that the micro movements of his face have contributed to an emotional contagion. Now, what's going on in the brain? And this is what's really cool. And this is kind of a neat analysis. This is why I wanted to share this with you. What we have done is taken his emotional ratings and used them as a regressor on her brain when she's watching his face. Woo-wee, that's pretty fun. And when we do this, we see, of course, this big Wernicke's area interpretation, which is really cool, and a lot of the dorsal lateral stream there. So one can say a lot more about this, but in this one sense, the exchange of emotion has a neurophysiological substrate that can be indeed associated with micromovements of the face. And this makes that point even more, where our facial cameras are actually recording the action units of each of the faces. And so these are like the many facial action units. And in order not to have to pick any particular facial action unit, we put them all together and we combine them in a principal components analysis. And the principal component then is used as the regressor. So here we have his face, the principal components of the facial expression of his face as a regressor on her brain. So she is looking at his face, which is well defined by the facial expressions, and we see loud and clear this lateral stream again, and the dorsolateral prefrontal cortex, and probably a bit of the motor cortex associated with indeed the facial mimicry. Her face is moving as his face is expressing. Again, some very, very interesting, I think, deep dive into multimodal approaches and also analyses to accommodate the modeling that can come from these computational approaches. So yes, the data are consistent also with hypothesis 3. So we have neural evidence for all 3 hypotheses for a real and live face effect: eye-to-eye contact, continuous processing of micromovements, and indeed dyadic communication of microfacial movements, both involving emotional contagion and facial mimicry. Well, all right, why? Why in the world do we want to know this? Why is any of this important? Why are these behaviors in that big other side of the behavior domain that we talked about earlier? Why is it important? Well, I'm going to give you 2 examples. One is because I'm going to give you an example in autism. And one is because these types of everyday behaviors are characteristic of medical diagnoses, and we can contribute to them. And the second one is going to be with the Zoom and our the new prevalence of remote communications. Okay, the first one's autism. And this is because autism is characterized by reluctance to make facial contact. Social difficulties are very prevalent, having to do with people unable to or unwilling to make eye contact and respond to other people's facial expressions. So it is kind of a natural to study with this type of paradigm. According to a recent report from Autism Speaks, that the diagnosis, the prevalence of autism is at least 1 in 36 children in the United States. So this is not a trivial matter. This is data that I showed you before. This is the TD typically developed participants with the eye-to-eye contact regressor. And our hypothesis is that in the case of autism, that actually we have 2 hypotheses too, that either this region will be hypoactive in autism, or that the autistic brain will process faces in a very different manner, so that there'll be an alternative neural circuitry. So this is going to be either depressed or that there's an alternative neural circuitry. And these are the autistic data that are acquired. And the answer is that we've claimed that there's evidence for a very distinct neural circuitry associated with eye-to-eye contact in autism: ventral for autism, dorsal for the typical. And when we compare the two groups together during the eye-to-eye contact task, again, we see the dorsal and ventral division. Very clear finding for the neurocircuitry of autism. One could say a lot more about this. I'm not going to. What I'm going to do is use this as an example to say we can learn a lot more about this type of diagnosis using live eye-to-eye contact and some very clever machine learning approaches with these data. So here, we've taken these data from this study, and I use as an example what I think is a small little entry point to a computational frontier for clinical application involving machine learning. And what we've done is subject these data to a support vector machine that trains on the diagnosis of TD versus ASD, which is great. We can train, this is our training. The training is pretty successful, but what's really cool here is that we do not train on the ADAS score. Now in autism, the ADAS score is a standard clinical measure of symptom severity. So what this says is that the higher, in this case lower, but the SVM score corresponds beautifully with the increasing severities. It's a very encouraging result. This is very new in our hands. We have this paper submitted. It is not yet published. I share it with you because actually we're very excited about these opportunities to use these type of data, these live data that we've acquired in live circumstances, and how perhaps useful that they could be in the hands of people that can apply machine learning to understand that the underlying neurophysiological substrates that we think might indeed represent a kind of biomarker for ASD. Here we are, this is the control where the eye to video condition, and we see no such correlation. So it truly is eye to eye. Okay, one could say more about this. The second and final example here is with something that we're all so familiar with, Zoom. Does it matter that the real live face is seen on Zoom or in person? Now look at the situation we have here. We've got two live faces. Here I've been saying it really matters that the face is live and not simulated, but here we have two live faces and they are the same face in this study. Does it matter? There we have no theoretical substrate that suggests that it should matter. They should be identical in terms of how the brain processes them. But I bet you're suspecting that it's not the case, and you're right. It does matter. I'll show you that in a minute, but I just want to emphasize that I think that this is sort of a new frontier that post-COVID practices have introduced us to. And this is a simple contrast in the in-person face and the Zoom face, same faces, exact same thing. Set this up with CIS, you saw the paradigm, and it matters a lot whether it's the real face. It's the face-to-face face or the Zoom face. And we could talk about the details, but the point is that I think that there's some major fertile directions here for studying it. This might even be related a little bit to the suggestion that I made earlier about loneliness, lack of connection between individuals as being something that is important for us to study with our new technologies. All right, last challenge, and then I'm off the page. How in the world are we going to fund this? We got to advertise this work. We've got to make it part of the mainstream here. And of course, we do have the Brain Initiative, and many of us have been funded by parts of the Brain Initiative as it's developed, and it's sort of a natural inroad for us to make our science, the science of the everyday world, part of the mainstream. So we do have an infrastructure, but what can we do as a group of investigators to assure that our funding base is going to be as solid as possible. And I have a few suggestions here, but I want to present this as the beginning of a dialogue for us to discuss during this next during the next couple of days that we're together. But there are relevant RFAs, requests for applications, and that we should apply for them, seek them out and apply for them for tasks and things that cannot be done with fMRI. When you submit your grants, check off the little box that asks for FNIRS expertise for review. You'd want to get as many people on those review panels that understand FNIRS. Otherwise, if you don't have people judging your grants that aren't familiar with FNIRS, things don't tend to go as well as they might. The collaboration across disciplines is really important, and advertise this in the innovation significance. Consult with the NIH program officers as every opportunity you get about the creation of new RFAs for new areas, such as the areas that we've been talking about. And then I think that this is also a matter for the Society for Functional Near-Infrared Spectroscopy to work with the NIH to secure as many opportunities for funding as we can for optical imaging or multimodal approaches. So these are all things that we can do to secure funding. I want to end with, this is, maybe I shouldn't, I've been advised not to show this slide, but you know, I'm not too good at taking advice sometimes, but I just want to share with you some of the comments that I've received. These are comments to me and my team from the NIH, from some of the grants that we've submitted. And the reason that I share them with you is because they're kind of cautionary notes and things that might help you avoid this similar pitfalls. One of them is that, oh, I love this one, this is the Dr. Hirsch's description of FNIRS as a new and emerging technology is overstated. Somebody didn't like that I said that this was an advanced technology and that we could do far more than what can be done with fMRI, I'm sure. But anyway, so the question, I guess the bottom line here is don't push it. The second one, the application lacks justification for the use of FNIRS over fMRI. This is a big point, and I hear this quite a bit. If you if you can advertise that this cannot be done with fMRI, it must be done with FNIRS. It helps, actually. You don't want to let people be confused about that. This is something that's very important to bring up because it is true. We just acknowledge it right from the get-go that the weaknesses of our technique include inherent constraints that FNIRS, which allows imaging of cortical but not subcortical activity. So we're looking at cortical stuff, not subcortical stuff. Just say it right up front to get it out there. They'll still come back and tell you that the technique is weak because we're not looking at lower structures, but nonetheless, that's the way it is. Not every technique can't measure all things perfectly. This is one that caused me a bit of consternation. The lack of retest reliability of the approach and methods undermines the scientific rigor and promise of the work, which may negate the overall importance. This caused me to write a whole long thing about how our technique is reliable, that it is replicable, and it is reproducible. And I think that in writing your grants, if you're any concern that you might have reviewers that don't think that this technique is as rigorous as others out there, that it's useful to write to anticipate that criticism. And the next three, I won't go over them in detail. They all criticize me for using big cis. And remember what I said, we're still in the laboratory. And so I think that this is a green light for all of us who are interested in the wearables. I'm interested in wearables too. I'm still in the laboratory. And we want to get these techniques out there. And here I have made statements about, I think that I'm studying real live dyadic interactions, and yet the reviewers come back and say, well, not really, Dr. Hirsch, you're not really studying real life interactions until you're out there in the real world. And I have to say that they're right. And so that's part of the mission for us going forward. So last slide here, wanted to just go back to our 4 challenges. What are our new questions? I hope I've given us a little bit of groundwork here to think expansively about what can we learn that we don't already know that sort of represents the real world of everyday life. How are we going to study them? And I know the next symposium is going to talk a lot about the technologies. So this is really interesting. How are we going to study them? What are our paradigms? The paradigms are extremely important. I don't know how we're going to get the smart glass on this wearable device. We've got a hemodynamic signal and we got to do something. So this is a pretty important question. Why do we want the answers? That means, how relevant are we? How can we make these answers? How can we make answers that actually contribute to how we understand the brain and how we treat it? And then how are we going to fund it? So those are the 4 grand challenges. I want to end by thanking my laboratory. It is no secret that that my laboratory is blessed with amazing people. And I just want to give a shout out to Adam Noah and Chen Zhang, computational neuroscience engineer, and Ray Capiello, who is our program lab manager. We have many students, postdocs, and people that support us, but this is my laboratory. And every time I've used the word I mean this I up here. Thank you.
There's plenty of time for discussion and questions. I just want to start off. It was very funny to see you say at the end, those last 3 reviewer comments, that people were complaining that your paradigms weren't real world enough.
I know.
Well, because the criticism I'm getting as I'm just beginning to kind of get this real world stuff into my grant proposals, and they're telling me you can't control the experiments if you're in the real world. You won't learn anything. Right. So
well, at least I haven't gotten.
So I'm not sure it's a green light.
No, I'm not sure that it is too.
Right.
Part of the reason for sharing that is that it is not a
sorry,
given. Sorry. That it's not a given that people are just going to give us a red carpet and say, gee whiz, this is really exciting. You guys go forth and understand the real world. This is something that we're going to have to really make an effort working with the systems that actually fund us.
If you have questions, maybe walk up to the microphone, just so people online can hear you. There's a lot of people online.
Even though.
Oh, and sorry, before Pepe, before your question, people online, please, you can type your questions online, and Manny here will get your questions presented to the speaker.
Okay, thank you very much for the nice talk. And in the most of the slide, which dealt with a gaze experiment, well, the activations were mostly on the right. And was there any reason for that laterality?
But here, because
That side.
I don't have to wander around. You know, I tend to wander, but I don't have to. This is really interesting, but the answer is yes, that most of the activation associated with eye-to-eye contact in a social real context have been observed on the right-hand side. No explanation for that. Sometimes I see an exception, and I think maybe you might have noticed that the real face with Zoom versus real face, it wasn't so right side dominant. So not sure about that, but we aren't the first to observe this. The Hoytel and Carter results in the scanner looking at observations of social activity also observed right side activity. I kind of think of it as a little bit of being homologous to Wernicke's area. Wernicke's area is the left side temporal parietal junction, which is interpretive, receptive. And the right temporal parietal junction is where the eye contact, the face activity is associated. So they're kind of homologous areas. One is for reception of faces and eyes, and the other for language.
Okay, I see. So is it that kind of the task dependent? For example, okay, well, if you are looking at an eyes and without any words, then probably if you have the verbal communication associated with looking at each other, then there may be the interaction would be more on the left side. Can it happen?
It is true when we do language experiments, we see the activity on the left, and the visual experiments when we see activity more on the right. But I haven't done the combined experiments to actually test that kind of massive parallelism of the brain, actually.
Okay, thank you very much.
Yeah.
Hi, thank you. When you were talking about real live versus video faces, I was thinking that the biggest theoretical difference there is the potential for cause and effect, like the other person might be reacting to me, and I could be attributing that. But then that should be the same for Zoom, like that should also hold for the Zoom faces. And I wondered if you think the Zoom versus real difference is something qualitative, or if you think it's a matter of degree, like if you could kind of get a fancy enough VR headset, high resolution, no lag, kind of centered camera, so you see eye contact being returned and all that, like if those would begin to converge.
I mean, that's such an interesting question. And of course, with the data that we haven't been able to sort that out. And if you noticed in the documentation on that slide, that paper is still not published yet. I think we've got this solved. It's been reviewed, it's in the hands, but that very issue has been the issue that's held it up, because we don't have an answer to that. If we could solve the problem of the technical issue of eye-to-eye contact in Zoom, would we get the same effect? I would like to think we would, but we haven't solved the technical problem because we can't do the same. The faces, the camera is always looking down, so we don't have the same eye-to-eye contact situation, and so the difference may indeed be due to that. But don't rule it out. I'll tell you, I've been fooled so many times. The brain is just so good at knowing when it's looking at a real face. And so I think it would take probably a pretty advanced technical solution to replicate the real face. But very good question. Thank you.
Hi, thank you so much for your talk. I'm Jessie. I'm a fourth year actually in David's lab, which means that I started my PhD during 2020. So a related question to what was just asked. I was wondering some of the specifics. I'm sure I'll find out when the paper comes out. But for Zoom, in thinking about my personal experience, learning over Zoom versus in person, there's a lot other body language and other ways to define the interaction. So I wondered, how far do you go in defining the differences between the interactions? Because there's so much else that's not controllable in naturalistic environments. So how do you go about that?
Well, I'm waiting for you to solve the problem. It's a really important question. That one of the things I didn't talk about here is reciprocity in interpersonal interactions. And this reciprocity involves not only eye contact, facial expressions, but also body movements and so on. So there's kind of, when people are interacting with each other, there's kind of a dance that goes on. You know, you go, I go, you go, I go. And it's a very natural thing that we do. It's not well understood. And so there are many parts of it that need to be studied and integrated into the theoretical framework that we'd like to develop. But it's all a very important part of it. And we're just the very, very beginning. So I think that your question really represents a very important future direction that we can kind of pin onto the framework that we're developing with what we have.
Thank you.
Great talk, by the way, but while the talk was pretty interesting, all of it was probably related to neural correlates, I guess. So I was wondering, when can we go beyond neural correlates to reverse inference as to like using the brain activation to sort of like characterize the nature of the social interaction itself?
But I'm not sure I understand. Are you talking about neurocorrelates and neurocorrelates with behaviors? So that my regressor technique.
No. What I mean is like, when can we go beyond, say, using brain activity to infer about social interaction, the reverse inference problem with social interactions?
When can we go beyond?
Like sort of like right now it's like, okay, I'm using social interaction and I get brain activations, right? But
social activation and brain activation.
Yeah, but when can I go from brain interactions to characterizing the nature of the social interaction, the reverse problem, yeah, the decoding.
Decoding the behavioral activity from the.
Oh, so you want to go the other way around?
Exactly, yeah.
Great idea. Go for it. It sounds like a good task for a good AI project.
Thank you very much, Joy. I'm Klaus. Just a short comment on the FNIRS cannot replace fMRI. I got the same comment for EEG, actually, even though I applied for mobile brain imaging. So I think it's really part of what reviewers are in the panel that decide on whether you get the money or not. And it's important to make clear that we move into a different direction. But on a related note, as the speaker before, so you've been talking about FNIRS, you've been talking about EEG, eye movements, EDA, GSR, basically lots of modalities.
Yes.
What you showed was only one modality alone in isolation. Did you ever converge data? How do you converge the data? How do you make sense of different timescales of the different modalities? And how do you try to get this kind of information together? Because I think the results are amazing, but the data is so much more. You could dive into that for the next 5 years.
Yes. This is a very important point, is that the data are so much more. We've just, I have a new postdoc that came into my lab, is from computer science, from AI, working now on trying to reduce the multidimensionality of these massive data sets. And he comes from a group of investigators that could care less about what the data is all about, could care less that it has anything to do with the brain, but really wants to understand how we can combine these many dimensions of data together to provide insight that we don't have. More power to them. I think it's got to be a good idea. I'm not quite sure where we're going with it yet, but we're going to have some fun with it.
Okay, thank you.
Hi, it's Sudan, a second-year PhD student at Boston University, also from David's lab. I was introduced to your work in one of his courses, in one of the papers, and really impressed. Thank you very much for this wonderful talk. So on the same line of Zoom interactions, and like Jesse mentioned, we miss a lot of body language, but we don't miss the facial interactions, although we miss the eye contact. I saw the eye box, and if the eyes were looking at other person's eye box in a Zoom, maybe you're still looking at the eye box, but the eye acknowledgement is missing. And so I was wondering, I know like there's no good technical solutions, but there some of the AIs like NVIDIA's never look away from the eyes AI that simulates as though the person in the video is looking at always maintaining the eye contact on the camera. That might not be the perfect one, but would anything like that just for pilot studies or anything like that help to simulate that eye contact acknowledgement and reciprocity?
I mean, in like a Zoom-like situation, I think that those are kind of goals that we have from an engineering point of view to actually improve the connections between individuals on a Zoom that allows real active eye-to-eye contact. That we don't have that yet. And we don't know if the difference that we see is due to the absence of that or to something else.
Got it. Thank you very much.
Thank you for a lovely talk. I really appreciate your candor and your leadership and sharing the challenges that you faced here in developing paradigms, getting funding, and I think that's so useful for so many of us here. Myself, I've come from fMRI, I've been working in FNIRS trying to develop these paradigms, and I find myself wanting to keep saying, well, maybe I do this experiment in fMRI and then do this in FNIRS, and you must have had those thoughts yourself as you face challenges. I'd just like to hear you expand on trying to write grants that have multiple, that are split between FNIRS and fMRI or otherwise.
Well, thank you for that. Yeah, okay. I didn't keep my promise. My general feeling is that if the experiment can be done in fMRI, do it. It's going to be better in fMRI if it can be done in fMRI. But the behaviors that we want to understand cannot be done in fMRI. You can't talk to somebody. And I don't care what these guys in fMRI tell you, oh yeah, we can get eye contact. Oh yeah, we can get contact. Forget it, they can't. So if what you want to understand has to do with live interpersonal interaction, you must use FNIRS or other systems that allow movement, interaction, dual brain computation, and so on. And so in writing grants, I try to make that point. Now, there are times when I have proposed and have used fMRI for calibrations. This is something that NIH study sections like a lot when you can document what you're doing in the scanner. So you can do a part of your process in the scanner and then connect it in some logical way. But I do that as more of a grantsmanship maneuver than something that I think is really important. But anyway, yeah.
Hi, Joy. Alan Reese at Stanford. So I completely agree with you about using fMRI for ground truth and validation for grantsmanship. The other comment I wanted to make for technical specifications, someone talked about the AI mechanism to try and maintain so-called eye contact during Zoom. It is really totally weird and spooky. And so it doesn't really work for the purpose. I don't know if you've ever seen that. It's just
tried it. I agree with you.
It's bizarre. It makes you think you're being monitored from some other galaxy or something.
Yeah, it's
Now, the last thing I did want to say is about Zoom interactions. We do have a couple papers out from our group, and I'll briefly mention this, and if anyone wants to have the references, I'm happy to send them, on Zoom interactions, one in the Journal of Neuroscience and one in Cerebral Cortex, on the difference between Zoom interaction and in person, and also expressing appreciation by Zoom or interaction. So Balters is the first author, Balters, Stephanie, out of Stanford. So if anyone's interested, we've done some initial work in this area and we thought it was fascinating.
I would like to follow up with that because it is fascinating. It's very important. And I think that it has, as we say, it has wings. It has a future direction in the kinds of work that we need, the kinds of problems that we need to solve with the kinds of techniques that we have.
Yeah.
Yeah. Thank you, Alan.
I think we have a couple online questions that Manny's going to read.
Okay.
Yep. So I have a question here from Bettina Sorger. So the question concerning real and live face effect. What if a face was presented in VR as the control condition versus the 2D photo or 2D movie? Do you think our brain can distinguish between a real face and a virtual reality face, assuming a high quality VR system?
We haven't done that experiment. I'd love to do that experiment. My general conclusion from the experiments we have done is that you can't fool the human brain. The brain is just very, very, very smart. So I would be very surprised if the VR could do that, but would love to see that experiment. It's a very good question. Thank you.
I'm going to just ask one last question, and then I'll let you guys go to your coffee break. So I assume all the results you showed us were group results, as opposed to individual results. And so I wonder if you could comment on the importance of, getting what's the science you can do in the everyday world with group results versus do we have to really push towards getting individual results?
This is a huge and really, really important question. And it's not just science in the everyday world, but in medical applications. Patients come in packets of one. And that is one of the things that I loved about the autism studies that I showed you. Those were individual patients. Each individual patient was coded by the SVM machine. And the individual was coded against his or her ADOS score. And so this was an attempt that we had made in order to classify, organize, and understand the neurocircuitry of single subjects. But this is extremely important. Most of our group are most of our studies have been in order to understand the underlying neurocircuitry that can be generalized across individuals. And yet, much of what we want to understand in applications are single people, single cases. And so adjusting our technologies for individual characterizations is really important. Thank you for bringing that point. It's really important.
Yeah, thank you, Joy. All right, before thanking her, just want to tell you we have 17 minutes for our coffee break. We will start the next session promptly at 10:30. We'll bring you in a couple minutes before then. So please go out, enjoy your coffee break, continue this discussion, and let's thank Joy again.